The other thing to think about in a laboratory setting, once you have diagnosed the infection, is how are you going to treat that infection? For bacteria, the way we treat bacterial infections is through the use of antibiotics or antimicrobial compounds. And we have a lot of antimicrobial compounds, lots and lots of antibiotics. But the problem is, and as we talk about specific bacteria, you'll start to see this. The problem is that bacteria have started to evolve mechanisms for resisting antibiotic therapy. They have become antibiotic resistant. And what that means is that normally we have a drug that would have treated that infection, and now that drug doesn't work anymore. And that presents a big challenge for how do we make this patient better. So those of you who are looking to work on the clinical side, you will start doing things like antimicrobial susceptibility assays. When you took Bio 120, you almost certainly did some sort of antimicrobial susceptibility assay. It was most likely something that looked like this, and it's called an Auger diffusion or disc diffusion or Kirby-Bauer assay. And the idea is you test your bacteria against known concentrations of antibiotics, and you see if the bacteria is still able to grow. If it can't grow, great, you can still use your bacterium, or your antibiotic, excuse me. If the bacteria does grow, even in the presence of the antibiotic, you have a problem and have to move on to a new compound. Another type of test you can do, which you likely did not do in Bio 120, is a broth dilution assay. And that can help you determine what we call the minimum inhibitory concentration. And that's basically the lowest level of antibiotic that we can use in order to clear a patient's infection. And so you do that on a plate, usually a 96 well plate, but this is, just an example of a type of plate. You have increasing concentrations of the antibiotics. So this would be the highest concentration here. And then you start to dilute, and then this column would be the lowest concentration. Here are the different antibiotics. So each row represents a different antibiotic. And if a well has a dot, that means the bacteria grew. So if we look at the first antibiotic, we see no growth. So that means this bacterium is sensitive to this antibiotic. If we look at penicillin, we see at the highest concentration it didn't grow, but at the next highest it did. So this bacteria is resistant to penicillin. And then we see with methicillin, ampicillin, and cefotaxime that it's sensitive. It only grows at very low concentrations of the drug. So we would use this concentration to treat our patient with methicillin. We would use a little bit higher concentration of ampicillin, and then we would use this concentration right here of the cefotaxime. So it's important to know not only which drugs can you use, but also at which concentrations. So these are some of the important terms when we're talking about antibiotics, antibacterials. We'll talk about broad spectrum and narrow spectrum. So broad spectrum can work against a lot of different kinds of bacteria, whereas narrow spectrum work against a more limited subset of bacteria. We even have some drugs that only work against one genus. So we can get pretty narrow. We have bacteriostatic and bactericidal. Bacteriostatic inhibits the growth. Bactericidal kills. And sometimes bacteriostatic is just fine because as long as the bacteria aren't growing, that allows your immune system to come in and clear the infection. We have the minimum inhibitory concentration. This is the lowest concentration of antibiotics that prevents growth. We also have the minimum bactericidal concentration. This is the minimum concentration that kills 99.9% of the bacteria. The MIC is a little bit easier to calculate, so we tend to do these types of assays versus the MBC, but both can be done using that same type of plate system. What we'll also talk about is the idea that when a physician thinks about what antibiotics they want to prescribe, they sometimes might want to prescribe antibiotic combinations. And this can be for a number of different reasons. Perhaps a patient has an infection caused by 2 or more bacteria. So you may need to prescribe 2 or more drugs to help the patient clear that infection. This can also help prevent the emergence of resistance. So if we treat an infection with 2 drugs, and at some point during the course of the infection, the bacterium becomes resistant to drug number 1, there's still drug number 2 to come in and kill them before they can get out and spread that resistance. And then we also have the idea of synergy. So that's shown here, number 3, a synergistic killing effect. And then it's defined right here. But basically the idea is with synergy, if you put 2 drugs together, they basically enhance each other's efficacy. So they work better together than you would expect. You have to be careful though, because there is the opposite concept of antagonism, where you would expect drug A works this way, drug B works this way, you put them together, and even if they don't enhance each other, at least they'll still, have their same efficacy. But with antagonism, what can happen is when you put drugs A and B together, they actually work worse. They almost inhibit each other. So you would want to test in the laboratory how the drugs would work together against different bacteria. The last term listed here is beta-lactamase. And beta-lactamases are going to be really important when we talk about antibiotic resistance, because these are enzymes that destroy the most common class of antibiotics, which are the penicillins, anything that has that beta-lactam ring. So the beta-lactamases have been a hugely important enzyme for bacteria to allow them to resist antibiotic therapy, but for us, because they make us have to find new ways to treat our infections. So when we talk about how antibiotics work, the reason that antibiotics work and the reason that we have so many more antibiotics instead of things like antivirals or antifungals is because bacteria are so different from our cells. Whereas fungi are already eukaryotes, so they're very similar. And viruses literally take over our own cells, so of course that's very similar. But with bacteria, they build a cell wall made of peptidoglycan. So there are a number of different drugs that will inhibit their peptidoglycan synthesis. They use slightly different enzymes to do DNA replication. They're homologous, but they're not exactly the same. They use slightly different enzymes to do RNA synthesis. Okay. They use different ribosomes to do protein synthesis. So they have different rRNAs and different ribosomal proteins. And they make different metabolites. So if we can take advantage of the differences, of which there are many, we can have antibiotics that work. So what we see is that beta-lactams, those antibiotics, inhibit the formation of peptidoglycan. And so as a cell is trying to reproduce, as it's trying to make more peptidoglycan, it can't. The cell wall begins to fall apart, and that leads to death of the bacterium. Unfortunately, we see a lot of resistance to beta-lactams. And resistance occurs by 3 main mechanisms. 1, the bacteria literally just pumps out the drug. So theoretically, the drug would still work, but it just can't get to a high enough concentration in the cell because the bacteria keeps throwing it out. Another mechanism is by modifying the penicillin binding proteins. So if the bacterium can modify the proteins in such a way that they still work to build the cell wall, but don't bind the antibiotics anymore, that works very well for them. Or they have those beta-lactamase enzymes that just cut up the antibiotic and prevent them from working. The class of drugs called glycopeptides, they also interfere with that bridge formation, that cross-linking of the subunits of peptidoglycan. And remember, the subunits of peptidoglycan are going to include the N-acetyl glucosamine and the N-acetylmuramic acid, and then those 4 peptides that come along with it. So glycopeptides work by interacting with that D-alanine terminus. So I want you to reflect on how you would think resistance could occur. We have a lot of different protein synthesis inhibitors. The aminoglycosides bind the 30S ribosomal subunit. That leads to either altered protein products or even early release of the ribosome, so they can't make the correct protein. So the bacteria can't make the correct protein, or it can't make the protein at all. So again, I'm going to ask you to reflect on how do you think resistance occurs. Tetracyclines also inhibit at the 30S subunit. They block tRNA from binding. So if tRNA can't come in, it can't bring the new amino acids, we can't build a protein. The resistance mechanisms are similar to aminoglycosides. We also have drugs that can bind the 50S subunit. So chloramphenicol binds the 50S subunit of the ribosome, which blocks protein elongation. We have macrolides that bind to the 23S rRNA gene, or rRNA product of the 50S subunit, and that also blocks elongation. But again, there are bacteria that have resistance to all of these. So for every drug we have, there's at least one bacterium that has resistance to that drug. Our quinolones affect DNA replication. They interfere with DNA gyrase or topoisomerase. So I want you to think about, what does DNA gyrase do during DNA replication? And so why would a quinolone affect DNA replication? So you need to think about that. Go back to genetics. How does DNA replication work? Specifically those enzymes. Rifampin affects transcription because it binds the RNA polymerase, not allowing transcription to occur. A drug like metronidazole actually causes toxic compounds to be produced, and those damage DNA. Metronidazole is kind of weird. It's one of those weird antibiotics that can actually be used as an antiparasitic as well. So when we start talking about parasites towards the end of the term, we'll see how metronidazole can also be a common therapy for a specific parasitic infection. And then finally, antimetabolites block various steps of folic acid biosynthesis. So we require folic acid. However, we get it from our diets. Bacteria are not able to absorb folic acid. They have to synthesize it themselves. So there are a handful of drugs that prevent folic acid biosynthesis. So the bacteria can't make folic acid, and therefore they can't survive. Again, here are some of the common mechanisms of antibiotic resistance. We have the production of enzymes. The beta-lactamases are the most common that we talk about. Barriers that prevent access to the target. Pumping the antibiotic out of the cell. Altering the antibiotic target. Producing excess of the target, so they basically outnumber the antibiotic. The antibiotic target is no longer needed for bacterial survival. So this is how methicillin resistance works. The bacteria that are methicillin resistant, methicillin binds to a particular penicillin binding protein. The bacteria that are methicillin resistant just make a completely different protein. Or bacteria enter a stage of dormancy in the presence of the antibiotic. Antibiotics really only work as the bacteria are trying to grow and divide. If the bacteria is just going to sit there and wait it out, they could win. Because what you have to think about is patient compliance. So if a physician prescribes you an antibiotic and tells you to take it for 10 to 14 days, let's say it's a 14-day prescription, but you start feeling better at day 6 or 7, you may forget to take your antibiotics someday, or you may think, well, you know what, I'm just going to hold on to this until the next time I'm sick. But what could have happened is the bacteria are just kind of hunkered down and waiting for you to stop taking the antibiotic, and then they'll come back and be resistant because they've already seen what you're going to throw at them. They've survived, they hung out, and now you're in trouble. So patient compliance is really key for some of these mechanisms of antibiotic resistance. Okay, the last thing that I'm going to show you is a video that was produced by Harvard. And it's really cool because we talk about antibiotic resistance, and really it arises through evolution. And we always think of evolution as this long process that takes, millions of years. But remember, with bacteria, something like E. coli, they replicate within 15 or 20 minutes. So for them, thousands or millions or billions of years is like weeks, months. Okay, so how quickly can antibiotic resistance evolve in a bacterium?
So what we ended up building was basically a petri dish, except that it's 2 feet by 4 feet. And the way we set it up is that there are 9 bands, and at the base of each of these bands, we put a normal petri dish thick agar with different amounts of antibiotic. On the outside, there's no antibiotic. Just in from that, there's barely more than the E. coli can survive. Inside of that, there's 10 times as much, 100 times, and then finally the middle band has 1,000 times as much antibiotic. And then across the top of it, pour some thin agar that bacteria can move around in. The background is black because there's ink in it, and the bacteria appear as white. First, you see they spread in the area where there's no antibiotic, up until the point they can no longer survive. Then a mutant appears on the right. It's resistant to the antibiotic, it spreads, until it starts to compete with other mutants around it. When these mutants hit the next boundary, they too have to pause and develop new mutations to make it into 10 times as much antibiotic. And then you see the different mutants repeat this at 100. And after about 11 days, they finally make it into 1,000 times as much antibiotic as the wild type can survive. And so we can see by this process of accumulating successive mutations that bacteria, which are normally sensitive to an antibiotic, can evolve resistance to extremely high concentrations in a short period of time.